Thanks for taking the time today, although it's your holiday,
Yeah.
a vacation, I've understood.
Yes, I'm crouching on the floor here.
Okay.
In the bedroom.
Okay, so today Jason Villagomez is joining us. He's the marketing manager at Nanian. Could you just once again give a brief overview of your application and your main objectives?
Right. So, we work in my group on a large number of transporters from the mitochondrial inner membrane. There are 53 of them in total. Actually, 52 are technically in the inner membrane. And they transport every component of the food that we eat across that membrane. So they fulfill an essential function, really, without the mitochondrion couldn't be. And some of the major functions that we work on are the transport of ADP and ATP across the membrane, and but also, for instance, the phosphate carrier. So the phosphate and ADP need to go over the membrane to synthesize ATP, and then ATP has to be brought back out. And this is, we have to transport our own body weight in those components every day. So it's a major transport function. But all food components are transported by these transporters. So whether that be inorganic ions, amino acids, fatty acids, vitamins, and for many of them, the mechanism really hasn't been defined. And I like to think that we made kind of major contributions to understanding how that works, but there are many mysteries left. And a big one is to explain how anions, which many substrates are anions, are transported against the membrane potential. It's about 180 millivolts. So, we've worked out that the transporter works as a monomer, and therefore you have to have two steps, one step going in and one step going out consecutively. And, how can you explain that? So a big issue that's outstanding is how do these carriers achieve that? And as I say, they do so for a huge variety of different substrates. So we concentrated recently, actually, with Andre on the ADP carrier, because it's such a well-studied thing. But those riddles exist in many of the other transporters. How do they actually achieve it? The other big problem that's outstanding, really, I think, where we could use this device is to understand aspects of proton coupling. So I think that's one of the big remaining questions in a general sense in bioenergetics. How do you couple the transport of a substrate to the transport of a proton, and what does that actually do? What is it for? What does it try to achieve? And people, would say, oh, it is to bring anions in, but the truth is you've got anion transporters that are not proton coupled, and you've got proton coupled anion transporters. So that's not the answer. So the question is, how does that work, proton coupling, and what does it achieve when those transport steps are used, are proton coupled? So that's another big outstanding biological problem, actually, to work on and to resolve. And in the lab, again, we work on the basics of it, to identify maybe residues that are involved. But to understand the process, you need to understand how charge is actually moved. And as all the preliminary stuff has shown, this is the way to do it. It's to unravel these processes, if you want, and to find the residues that are involved, to understand their contribution to this process. And I think, and I think we're perfectly capable of really completely dissecting that, because we have these proteins in our hands, we have expressed them in a recombinant way, and we can purify them, stabilize them, we can reconstitute them, and put them therefore back into liposomes. So we're in a really good position to use the machine, because we can, do the fixing and then measure these transporters and the charges that they move. And some are electroneutral, some are electrogenic. So what makes one electroneutral? What makes one electrogenic? Again, with mutagenesis, we can work out which residues are involved, the mechanism, how they achieve these things, these particular steps. And then I think the big other one that's outstanding, which is, a large number of transporters, probably about a third, I think is fair to say, of the 53. So that's a huge number. Well, we don't even know what they do. And it's very difficult to get a starting handle on this. So we know what substrates need to be moved, but we don't know which substrates are transported by which transporter. And it's a bit of more of a potluck approach, but I think, in principle, we should be able to give those substrates to the transporters cold, which is what makes the device that you have so useful, because you could just buy those compounds and see if there is a charge movement occurring in a single step, for instance. That would be a great thing. So we thought we could use maybe to identify some of the remaining ones, or to look at the ones which I would call are disputed. There's a lot of claims for some of these transporters, what they do, but I look at it and think something is wrong here. Yeah, either they haven't identified the right substrate, or they haven't provided any proof for it, or it's disputed, where people say it's either this one or that one. And so there's no definitive transport assay has anybody's ever done to actually show it's one substrate or the other. And one bottleneck is there maybe to get radioactive compounds. That's another, it's hard to get for some of these, for some of them. Or you have, like, for instance, if you take pyridoxal phosphate, there might be 6 different versions of it, slightly different, either the amine or the acid, and then you don't know which one it is, and then you can't use your radioactive thing. So for some of them, I would like to have a look and see which substrate is the more likely candidate, and then we could take it forward. So there's a huge number of applications, and that's just the family that I've just mentioned. There's other families, where there's disputed substrate specificities, where there's absolutely no mechanism. Let's say the pyruvate carrier, there's no mechanism for it at all developed, because nobody's managed so far to actually get the purified complex in hand, but we have managed to do this, and so we are in the best shape to define the mechanism. So that's another activity in the lab. Okay, so that's roughly the kind of things that we're interested in doing, and are in the position to actually do. So,
thank you, Edmund. I mean, when I hear you talk about your science and your science project, even I would like to go back to the lab.
Yes, well, you're welcome.
Yeah, so therefore,
we have too many projects. But those are fundamental to life. We're not also talking in terms of, this is, every one of them does something really important, without which the mitochondria doesn't work, and if the mitochondria doesn't work, the cell doesn't work. So they're all fundamental problems, and they're fundamental for the function of humans in general. So, and we work on the human proteins, so also in terms of importance there, this directly translatable into disease patterns and why some diseases occur and so forth. Sorry, another round of propaganda.
So we're happy to announce that you have received the SURFE grant.
Oh, that is excellent news. I'm so pleased. And I've already felt sorry for Andre that we launched into one experiment after the other. So
no need
finally, and so that we can get our hands in action there. Of course, we need your guidance, but that's really excellent news. I'm very, very pleased.
Yeah, congratulations. We're very pleased too.
I'm extremely pleased. And of course, that means we'll have to buy stuff as well. And so I'm sure that, and I have already said that I would make an effort. I'm planning to write this ERC grant to extend this, because as I say, the number of problems we have, certainly warrants to. So I also, my side would pledge to see if we can get something more permanent. But this is great. So, and I think, and also to maybe get a foundation for that grant would be great, by showing that this can be done, because I think it's a unique opportunity, for really unique opportunity for us. So thank you. Thank you very much.
Yeah, thank you for sending your application. And as I said, I think I speak for us all. We're very excited.
Okay, good. Great.
Comes out of this project.
Thank you. Thank you. So now the question is when? We can't wait. We can't wait.
Yeah,
So
exactly.
when would it realistically be done? Because then we can start planning our stuff and you know, work towards an optimal use of it as well.
Yeah.
So I appreciate this is about 6 months.
Yeah, unfortunately, all of our systems are sold out right now.
Right.
But we have another batch in production.
Yeah.
So they will be ready to go towards end of September, I would say.
Right. Okay, cool.
But Andre, I suppose you will reach out with more information.
Yeah. Definitely, yeah.
Okay, be great, because we do need your expertise really to get us going.
Yeah, of course. I mean, I will help you with installation and training and setting everything up, and then I will also support you in planning the essays, and we can spend time to discuss the results. Of course, I mean, it's a new technique, so yeah, we will definitely keep on talking about everything.
Right, because I mean, I'm very excited with what we've done already. I mean, our discussion last week, I mean, it was just, was it last week? Yes, it was last week. It was amazing, I have to say. There's the prospect of really completely untangling this incredibly complex mechanism into concrete steps. So yeah, so I'm very keen. I'll tell the lab so they can start gearing up. I think there's already a queue, I'm telling you. Already a queue for this device. Because I do think a lot of projects really could need this. So, yeah.
Yeah, I also think so. Yeah, you will profit a lot from that.
I think so. I think so. And of course, we'll publish together. That's the other thing that I will pledge to. Yeah.
Yeah. Cool.
Okay.
Sounds good.
Well, very good news. Thank you. I'm so pleased.
Yeah, so I guess that was a time well invested, even though it's your vacation and holiday today.
Yeah. No, I was so keen. I'm happy to give up my vacation.
But we'll let you continue, and once you're back, we'll start to planning for.
Yes. Okay. Well, thank you very much. I'll pass on the good news to the lab. I think they'd be very excited.
Wonderful.
Yeah. Okay. Thanks.
Cool.
Thanks very much.